So I'll just start by introducing Michelle Moerel. So Michelle is an assistant professor in the Faculty of Science and Engineering at Maastricht University in Maastricht, Netherlands. She combines ultra-high field MRI and computational modeling in order to investigate auditory processing and plasticity in the human brain. She completed her PhD in cognitive neuroscience at Maastricht University in 2013, where she worked on auditory and subcortical processing using fMRI. And next, she moved to Minneapolis, Minnesota in the United States to complete a postdoc at CMRR, and she concentrated there again on auditory processing using ultra-high field MRI, both structural and functional. In 2015, she returned to the Netherlands where she continued her work at the Maastricht Center for Systems Biology and the Faculty for Psychology and Neuroscience at Maastricht University. And she was promoted to assistant professor in 2017, also within the Center for Systems Biology and FPN. And today we'll hear her talk about using laminar fMRI and computational modeling in order to find detailed examinations of auditory processing in humans.
Thank you. I hope you can hear me, and I hope that you'll be able to hear sounds as well, because I have lots of sounds as this is an auditory talk. Let me know if anything is inaudible. Yes, I'm switching modalities here, so I will tell you about auditory processing. And I first want to, because I assume most of you are much more familiar with visual cortex instead, I first want to tell you why we would care about auditory cortex, combining that with laminar fMRI. So from animal electrophysiology, we know that within the superior temporal plane, and in particular the Hassel's gyrus, which is this bump that you can see here, there is an interesting organization in the cortical sheet. Now, the auditory cortex is a little bit thicker than the visual cortex. That's good for us. It's just under 3 millimeters. And we know that feature tuning is relatively stable orthogonal to the cortical sheet as it is in visual cortex, and that there is an interesting laminar organization as well. So there's a larger there's a more complex feature tuning in superficial primary auditory cortex compared to middle layers. For example, more neurons respond to combinations of sound aspects, and fewer neurons respond to pure tones in superficial layers compared to middle layers. Now, this is all coming from animal work. So far, in the human, little was known, so we set out to explore these kind of organizations with cortical depth-dependent functional MRI. And I will present you some published work first, because they really serve as a background for ongoing studies. And these are, this is data that I collected at CMR actually during my postdoc, where we measured brain responses to a lot of natural sounds that include, for example, speech and animal sounds, tool sounds, and we collected those brain responses with two contrasts, gradient echo EPI and 3D grace, and then used functional MRI encoding to fit and compare models reflecting our hypotheses on what voxels might be doing. And we based these hypotheses on what we saw in lower resolution data. So we first employed a frequency model where we assumed that the response on a voxel would be well explained by the frequency selectivity, the frequency preference of that voxel, and then compared that to a second more complex model where we added two features. So in addition to preference for frequency, we hypothesized that voxels would also be tuned to temporal modulations and spectral modulations. Just to give you a bit of insight on what those sound like. So in this knocking sound, you see temporal modulations. These are the vertical stripes in the spectrogram. Music sounds will have temporal spectral modulations. So these are the horizontal stripes in the spectrogram. And many natural sounds have combined spectrotemporal modulations as well. such as speech, and you see these diagonals in the spectrogram. So because of this combined spectral sample modulation content and many natural sounds, we added these three acoustic features in a joint manner, such that a voxel could, for example, be tuned differently to temporal modulations for different frequencies. Now, the first results that we observed very much confirmed what we saw in lower spatial resolution data. And I'm showing you here a prediction accuracy score for the two models. And this reflects how well the predicted responses to a sound correlate with the responses that we actually measured, and specifically if they correlate best to the actual sound that we predicted, right, better than the other sounds. And a score of 1 would be a perfect prediction or picking up the correct sound, and 0.5 corresponds to chance. And you see here that both models perform above chance, but that the spectral sample modulation model outperforms the simpler frequency model. But now with this data at a 0.8 millimeter isotropic resolution, we could go and zoom in a bit more. And specifically, we did that separately for primary auditory cortex and non-primary auditory cortex, which we separated through a myelin-related contrast. And these are two star-weighted measurements. So I'm plotting now, again, prediction accuracy on the y-axis, but now on the x-axis, you see deep, middle, and superficial gray matter. I'm plotting 9 values, but the statistics that I'm reporting are done on 3 values only. And what we observed was a significant interaction between model and cortical depth in primary auditory regions, such that only in superficial layers, the spectral temporal modulation model actually outperformed the simpler frequency model. But in non-primary auditory cortexes, this was not the case. There was a stronger or a higher prediction accuracy for the spectral temporal modulation model throughout cortical depth. Now, this is gradient echo API data, so of course we must be careful with this interpretation. But what we're actually doing here is feeding the same data with the same biases into both models. And what I'm not showing you here, but you can find that back in our 2018 or image publication, where we directly compared model performance between the gradient echo and the 3D Grace data set, is that if you look at the performance of one model, so prediction accuracy for one model, then that and gradient echo data is very much biased towards the surface. And you can actually see that pattern back in both of the figures that I'm showing you here. But if you look at the difference in model performance between the two models, then the conclusion you draw from that is actually the same in the two data sets, in the gradient echo data sets and the Grace data set, which makes us have faith in that we can interpret this pattern. And we interpret it as follows, that in line with what was already known from animal electrophysiology, it seems to be the case that an interesting transformation is taking place in superficial primary auditory cortex. And this could very well reflect what is also seen in animal electrophysiology, that you get an encoding of specific feature combinations, which could serve as a first step towards creating towards sound abstraction and ultimately then sound perception. Now, from the gradient echo, from the 3D Grace data, we computed preference maps as well in order to examine how stable they would be throughout the cortical depth. So I'm showing you here data from one individual. For display purposes, these maps are smoothed per cortical depth. They're not smoothed throughout the depth. Let's say the cortical depth runs here from white matter at the bottom to CSF on the top. And you see the preference for frequency, temporal modulation rate, and spectral modulation scale. And you can already see from these maps that there are regions where preference is relatively stable throughout cortical depth, but also many regions where this varies. And we quantified that stability by comparing the variance along the cortical sheet to the variance orthogonal to the cortical sheet. So every location on the cortical sheet got a value. And then we assign significance to that value by doing permutations, and based on that, creating a null distribution. So in this way, we got a map for each individual hemisphere and each individual feature of which locations had a higher stability of feature preference throughout that than expected by chance. And we could then do group summary statistics on these values. So what I'm showing you here is the percentage of columnar locations in primary and non-primary regions for the 3 features across individual hemispheres. And the first thing that you can see is that the stability of frequency and temporal modulation rate is higher than that of scale, and that actually matches what was shown in cat auditory cortex. And the second thing you can see is that primary auditory cortex has a larger percentage of kind of significantly stable preference throughout cortical depth than non-primary regions. And that was something that hasn't been explored in animal electrophysiology yet. So that's a unique human fMRI finding. The other thing that we observed is that we basically saw no locations that were columnar or significantly stable for all three features. And that suggests perhaps a coding strategy where the preference to certain features is kept stable throughout a cortical sheet, perhaps acting as an anchor almost, along which you can then have interesting computations taking place, let's say, in the feature that is being varied. And if this indeed holds, then that would mean that you actually have a different computation in the auditory cortical microcircuit for different locations on the auditory cortical sheet. So with this, we kind of examine the results that were there on the organization of the auditory cortex coming from animal electrophysiology. And the next question to ask then is what does this mean, if it means anything? What is this organization good for? And that is something that I cannot give you an answer to yet, but is something that I'm very interested in exploring. One direction that we're taking now is keeping in mind that this acoustic feature tuning is actually extremely flexible and that it can very rapidly adapt to changing task demands. So it may very well be that we can only get a good view on the function of this organization when we emerge the auditory cortex in something meaningful, right? If we let it do some meaningful task. So what we're now exploring is feature-based attention. So that is what I would like to show you in the rest of the talk. These are still preliminary results. So I think for me, it's also good to use this opportunity to get feedback from your side. To introduce this topic a little bit, I want to show you what we know about auditory feature-based attention, and in the auditory field, that's basically frequency-based attention. So most studies, they direct attention to a specific frequency range. Again, from animal electrophysiology, and this is now mostly work in ferrets, there's a lot of evidence that frequency preference and selectivity can change very rapidly in neurons, from neuron to neuron also differently, when you let these ferrets attend to a specific frequency. And this is really in the order of seconds that these shifts can happen. They occur in primary auditory regions, but are much stronger when you go up in the auditory hierarchy. So they're much stronger in secondary auditory cortex and belt, and even stronger in parabelt, so tertiary auditory cortex. And the exact changes depend very much on the exact task and the reward structure. And that you see in this figure here, that is coming from a paper by Stefan David and colleagues. And what you see here is data spectrotemporal receptor fields from two neurons in two different tasks. So if you look at the top, what you see here is the estimated SDRF of this neuron as passively measured. And then in the second column, you see what happens to this spectrotemporal receptor field as the animal attends to the target frequency, which is indicated by the red arrow. And in this case, it's an approach task. So what this means is that the ferret, if it hears this target frequency, it should lick a water spout and get some water as reward. And what you can see here is that in this approach task, actually, there's a suppression of the attended so the target frequency with behavior. Instead, if you change the task to an avoidance task, then you see the complete opposite result. So you actually see that there is a boost at the site of the attended frequency. So from the animal research, you get a very dynamic kind of mechanism, depending very much on the structure of what the animal is doing exactly. Also depending very much on what frequency the neuron was originally tuned to. So these changes are largest in neurons that were already tuned to frequencies that were close to the target frequency. Human fMRI is showing a very different picture. So human fMRI studies actually very uniformly show stronger responses to attended sounds. And they do not show population receptive field changes, although so far actually the studies that are out there did not test these population receptive field changes very much. So they generally either did not test at all or did it in a very coarse manner. So we set up to kind of merge these kind of lines of research and designed an fMRI study that was in experimental design very close to the animal electrophysiology and kind of going away from what was typically done in human fMRI. So this is a 70 study. I'm sorry to disappoint you, it's not a laminar study yet. We're getting there, hopefully. But we wanted to get a first kind of idea of what we were dealing with, where we had to look, and what kind of changes we would see as well in order to then optimize the design of our future laminar work. In 8 participants, we collected 5 sessions of data, and 3 of these sessions actually are from a study that is already published 2 years ago. And we recalled these participants and had them take part in 2 additional sessions. In these 2 additional sessions, sessions 4 and 5, participants heard artificial ripple sounds sequentially presented with natural sounds. And that sounds something like this. So these ripple sounds are the woo, 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 woo. And they were used to manipulate frequency-based attention. The natural sounds, responses to the natural sounds we use for PRF mapping separately for the different attentional conditions. We had participants detect noise bursts, that very short noise bursts that could occur only in the ripple sounds, so either in the low or high ripple sounds, 300 hertz and 4 kilohertz, and we varied the chance of the bursts occurring. So they either were much more likely to occur in the 300 hertz ripple sounds or in the 4 kilohertz ripple sounds. And that indeed successfully manipulated attention. So these are behavioral data from a soundproof booth, where you see that participants respond faster to the noise burst when they occur in the ripples of the attended sound frequency. So you see here that they respond faster to 300 noise bursts and 300 hertz ripples when they attend the low frequencies, and faster to noise bursts in the 4 kilohertz ripples when they attend high frequencies. In the scanner, a similar pattern was observed, although there the interaction term was not significant between attention and ripple frequency. Then we started to look at the fMRI data, and we had an extremely surprising finding, which is that contrary to what was so far reported in fMRI studies, we actually observed weaker responses to the attended ripple sounds. And this was especially there in belts, so secondary and tertiary auditory cortex in the right hemisphere. And we started to look into this effect by examining the responses to the natural sounds to try and explain this. As I already mentioned before, we did that through PRF mapping based on the estimated responses to the natural sounds. And we followed a permutation-based model grid search for several BLAS designs with PermGS, which you can read much more about in a publication last year by Agustin Lage-Castellanos. And what that means basically is that we estimated for each voxel the frequency preference and the selectivity of that preference as the mean and size of a one-dimensional Gaussian. So far same as standard PRF mapping, but the difference is that we now base the selection criteria on the probability of selecting a particular combination of mu and sigma under chance. And that is not uniform. Selecting more narrow Gaussians, so smaller sizes, is more likely in voxels that have a low signal to noise ratio. And this modification of the standard PRF mapping procedure accounts for that bias. We could reliably fit or or do perform PRF mapping in the different participants in the different conditions as well. So what you see here is the correlation between predicted and measured responses to the sounds, computed in each voxel and then averaged across voxels and across cross validations. And you can see that these values are reliably above 0. We then looked at the estimated maps, so the preferred frequency as the mu of the Gaussian and the tuning width as the sigma of the Gaussian. And I'm showing you here single participant results. I'm not sure how familiar you are with looking at these auditory maps, but I can tell you, and you may have to trust me on this, that this is what they should look like. So what you see here is in the tonotopy maps, so this is maps of frequency preference, a blue, red, blue pattern crossing Hassel's gyrus, right? And you see that here, 2 blue, red, and blue. Hassel's gyrus is indicated by the white dotted line. In the tuning width maps, what you see is a purple bluish region indicating more narrow tuning on anterior Hassel's gyrus, which we can interpret as the location of primary auditory cortex. The other thing that you may notice is that the maps are actually very stable across conditions. So you see the baseline data of the previous experiment on top, followed by the attend low and attend high condition. And this is reassuring, especially given the fact that this data in many participants was actually collected 1 to 2 years apart from each other. These are the group maps averaged across the 8 participants. Nothing surprising here, except that you can see what you should see, the pattern that you should see in these maps a bit better. We then went on to look at histograms of these maps to see if we could observe any differences with attention. And I'm showing you here on top maps of histograms of best frequency in core, so primary, belt secondary, and parabolic tertiary auditory cortex. And in the second row, you see the tuning width, again for the 2 conditions, so attend low and attend high. And we did not see any obvious shifts, any obvious changes that differed between the 2 conditions. So nothing to explain our responses to ripple sound. So what we then did is to, inspired by the animal results, look in those voxels and specifically that had a best frequency that was close to the attended one. So what I'm now showing you is again this response to this lower response to attended ripple sounds that you already saw. But then on the right, I'm adding a graph where on the x-axis there's where basically we group the voxels depending on their distance. The distance from their best frequency to the attended target frequency in octaves. In black lines, you see the attended response to attended and non-attended ripple sounds in dashed and solid lines. And in blue, you see the difference between these 2 lines. And what we observed was that the difference between responses to attended and non-attended ripples was actually only significant in that cluster, that bin of voxels that had a best frequency closest to the target frequency. And we also observed a significant interaction between condition, attended and non-attended, and distance of the best frequency of the voxel to the target, such that actually as you go further away in your best frequency from the target frequency, the difference in response to attended and non-attended ripple sound gets smaller. We looked further into these voxels to see if we could find differences in PRF as well, and we observed a difference in tuning selectivity. So a little bit in the belt, but only significantly so in the parabelt, we observed that there was an effect of attention on the tuning width of only the voxels that were close to the, whose best frequency was close to that of the target. And what this then, how we interpreted this is as follows, that we seem to have, with frequency-based attention, a narrowing in the tuning width in best frequency match voxels, mostly in parabolic regions, and that in those voxels also we get a lower response to ripples. And that may be explained by the fact that our ripple sounds are not pure tones, right? They have a bandwidth, they actually are 1 octave broadband sounds, let's say. So you can imagine that perhaps if tuning width decreases, that may result in driving basically less voxels and ultimately in a lower response recorded in the fMRI data set. While that kind of fits within our study, it still leaves completely unexplained why this is so different from previous fMRI studies. And so we looked a bit more into that, and I think at least the best explanation I can come up with so far is the large difference that there is in experimental design. So all of these previous fMRI studies, they employ a paradigm where you have both targets and non-target frequencies present simultaneously. So they are generally designs where you, for example, have streams of low, middle, and high frequencies, and then a participant has to attend one of them and suppress the other two, for example. That is very different from what was done previously in electrophysiology, also very different from what we did, because both of those designs are sequential presentations, right, where you have different sounds occurring and the target occurs in isolation, let's say. So what this hints at is that the difference, that very different mechanisms may underlie these different tasks, these different experimental paradigms. So another thing that I would briefly like to show you also as an outlook is that we've been doing a different kind of modeling. So this is not encoding or PRF modeling, but this is mechanistic modeling. We adapted and this is mostly work of Isma Zulfikar, whose publication you can see reference at the bottom. We adapted a Wilson-Cohen cortical model for representing auditory cortical processing. So specifically, this is a firing rate model where the firing rate consisting of a set of coupled differential equations representing an excitatory neural population and an inhibitory one, and where the firing rate of these populations is modified by external input, but also by self-connectivity and cross-connectivity, shown here by the different W parameters. And we modified this model by having sounds pass through a stage of processing, a filter bank, which represents peripheral processing. And you can see this basically as a modified Fourier transform, and then passing the energy of the sounds at these different frequencies that are coming out of the Fourier transform to different excitatory units, and thereby replicating the tonotopic axis that is the main organizational principle, as far as we know, of the auditory system. And we've shown that doing this and then afterwards fine-tuning the parameters a bit based on animal electrophysiology, we can actually replicate several findings from human psychoacoustics with this model. And what we're currently working on, and when I was hoping to show you results today, but I didn't manage to get them solid enough to show them, is that we are planning on or modifying basically the connectivities to change the tuning width, as we see that also in our fMRI data, and then seeing if that can explain the changes in response to the ripple sounds that we observed. Isma has also worked together with Martin Havlicek in linking this Wilson-Cohen model to fMRI responses through PDCM, which would allow then testing changes in which changes in neural dynamics could also not underlie our fMRI findings. So yeah, this is perhaps something that also could become a discussion point. What the goal of all of this work is to extend it to a cortical depth-dependent study. So in order to explore the origin of these PRF changes, but also maybe to get them more precise, because we know also from some previous work that, and also of course visual cortex, that we should expect a difference, a cortical depth-dependent difference in attentional effects. So we may actually, by going cortical depth-dependent, also increase our sensitivity to pick up these effects that may be averaged out now that we're going across the whole cortex. Knowing that we have a different stability throughout the cortex for different acoustic features, it would be very interesting to do this for different features, not just frequency. And we have extended the behavioral paradigm and now know that this noise burst detection task also can work to direct attention to either slow or fast temporal modulation rates. So that is a dimension we can add to it. And then ultimately, that may allow exploring if there's a relation between the effect of feature-based attention, cortical depth dependently, and the stability of PRFs, right? So if we have locations which are stable for example, frequency, and other locations that are stable for temporal modulation rate, can we observe a difference between these locations of cortex in the effect of feature-based attention? And ultimately, if we can actually extend our computational modeling approach to a laminar resolution, we may actually get at the biophysical underpin, neurophysical, physiological underpinnings of this. So that is, I hope to be able to show you results of all of these goals at some point, but it's still quite a lot of work to be done. However, in what we see already from our non-laminar results is kind of where we are restricted, right? We definitely need to, I think, get a data set that includes secondary, tertiary, and ideally also primary auditory regions. But as we're going to be interested in PRF changes, gradient echo EPI really may not be the way to go, which puts quite some restrictions on what we can and cannot do. Yeah, a lot of work to be done yet, I think. With that, I would like to thank a lot of people that were involved in various aspects of this work, and thank you for your attention, and I welcome any questions, any discussion.